Okay, yesterday then we had a look at the sort of overview of the nervous system. We looked at the component parts of it. We broke it down into sensory receptors, of which we had quite a selection there. We had the nerves that do the connections, so we had different types. The somatic ones that give us sort of conscious control. The autonomic nerves, which are actually more like the endocrine system. They are very much homeostatic. And we will come back to the autonomic nervous system. All right, those will be our sort of example there. Nerves, as opposed to neurons that we're looking at, nerves are bundles of neurons. So you will actually get sensory nerves, motor nerves made of sensory neurons, motor neurons. But you know, we can't cover everything in the depth that it would be nice to. But we'll be doing the autonomic nerves because they fit in well with our general sort of homeostatic approach to all this. And then the central nervous system, we're going to look at the brain in minimal detail, and we'll have a look at how reflexes work as well, because those are quite important. Now, from there, we went on to look at the structure of some example neurons, and we have there the labels that go on the diagrams in your handouts. So we've got the sensory neuron on the left, the motor neuron on the right. They've got a lot of things in common. They've got the cell body, although it's positioned differently. If it's part way along, then you're looking at a sensory neuron. If it's on the end, it's a motor neuron. The cell bodies, of course, are the same content as you'd find in any other cell. So your cytoplasm and the nucleus and all the membranes and mitochondria and stuff. Although you have a lot of mitochondria throughout the nerve fibres. We'll come back to that particularly because one of the processes, of course, is active transport. Remember writing sodium-potassium pump in your assignments. If you didn't, it's too late now. But the sodium-potassium pump, of course, for active transport uses energy. That energy comes from mitochondria. So they're going to be full of them. The differences between them really are only in the position of the cell body. Otherwise, you've got all the same parts. These fibres are named differently depending on whether they're going into the cell body or coming out. So dendrons and dendrites are what we should have going into the cell body. The axon comes away from the cell body, although you will tend to find the word axon used as a very general term for nerve fibres anyway. The other one that we had down at the bottom was our multipolar, relay, intermediate, whatever else I'd put there, neuron, which is found in the central nervous system and is very, very much responsible for creating memory pathways. And you know, it's the growth of the little fibres coming out of these nerves and making connections with other cells that forms memory. So whatever we're learning, it's about these cells interacting. These don't have myelin around the outside of them. Those do up there. And our next job then is to look at how these work. Now, one thing I haven't put on there is the comment that they are described as excitable cells. Nerve cells, neurons, and muscle cells are the two examples of excitable cells. So excitable means that you can actually generate a response in them that by prodding them in some way. And, you know, it may be shining a light on them. It might be receiving an impulse from another neuron, that you can actually create a response in the cell. Now, the nature of this response is, well, it's what we call a nerve impulse. And it's electrical in nature, but it's not electricity. Okay? Now, that means we've got to understand a little bit about what electricity is, because there is this feeling that nerves sort of carry electricity, but they don't. Electricity is the flow of electrons, usually through a metal, isn't it? All right, now that overlaps with your chemistry assignment in describing the elements in Group 1, doesn't it? Group 1 elements, they're all metals. They're described as the alkaline metals. And one of the characteristics of metals is that they have small numbers of electrons on the outside, and when they bond together in a lump of metal, all those electrons are actually delocalized. They're not round the atoms anymore. And so if you've got a lump of metal and you push some electrons into one end, other electrons will fall out of the other end. That's called electricity. Right? That's what electricity is. It's the movement of electrons through metals. You don't get it with other substances, but because the electrons in a metal are delocalized, and that's one of those features you should have in that section. So hopefully that bit's nearly done now. But the ability to conduct electricity is because of those loose electrons. So it's the movement of electrons. Now, what do you get in a nerve cell then? Well, it's not that. It's not the movement of electrons. But, of course, you can actually simulate the effect of a nerve impulse with electricity, can't you? You know, you can connect a muscle up to an electrical stimulus, and what happens? It contracts, yeah. Yeah, the muscle will contract if you provide an electrical stimulus. There's a whole product range based on that, which is, never heard of Slender Tone? Yeah, the twitchy belts, you know, that you put to make you know, the slimming effect. What's happening there is it's just taking away the need for your nervous system to make your muscles contract, and it just does it over and over and over again by sending small electrical impulses into the muscle that cause the contraction. They do work, yeah, yeah, because they are making the... No, a TENS machine is completely different. TENS machine shouldn't make your muscle contract. If it does, you've got it in the wrong place. A TENS machine is supposed to be positioned over a nerve so that it will actually send a little electrical impulse into the nerve, which actually interferes with pain signals. So, and why I'm not wearing one at the moment, I have no idea. I have two, and I have a pain in my back, and I just keep forgetting. But yeah, TENS machines work very well, but on, you know, on the understanding that electricity is similar to a nerve impulse, but they're not the same. So a nerve impulse, then, we've got to separate this out. It's not a flow of electrons, but it's something that has the same sort of effect, and it's about electrical charge. Now, electrical charge comes in two forms, really, positive and negative, and we've met that, haven't we? We've met the idea of positive protons, negative neutrons. We've met the idea of polarized bonds or polar bonds, completely polar molecules. And this is about the distribution of positive and negative charge. Now, we're going to do something similar with nerve cell membranes. And what we're going to look at with a nerve cell membrane is how you can set up a difference in charge between the inside of the cell and the outside of the cell. Now, when you've got a resting nerve cell, a resting neuron, it has a difference in electrical charge between the inside and the outside. And you think, well, how do we know this? Oh, it's simple. You just get little microscopic electrodes. You look down a microscope at a living nerve cell. You put one electrode inside the cell, another electrode outside the cell. You connect them to a voltmeter, and it tells you what the difference in charge is inside and outside the cell. And they can do that quite easily, as it happens. So, you know, measuring this difference in charge inside and outside the cell can actually be done physically, you know, quite relatively straightforwardly. So it's a matter of how we set up that difference, and so that's where we then need to sort of overlap with what we did in the last unit in terms of transport processes. And the first thing that we're going to do then is to set up what we call the resting potential in a cell membrane, and that's what you've got when the cell isn't conducting an impulse. All right, so I'm going to write this down on the board in a series of steps. Then I will show you a numerical way of illustrating it, and then I've not actually managed to track down a really good animation that covers the whole thing at the moment, but we might recap back to active transport for a start, because we did have quite a good animation that showed that sodium-potassium pump quite clearly. So we start with that, and then once we've got it clear that that's the resting state of the membrane, we'll move on and look at what happens when there's some sort of stimulus, that then creates the impulse that's going to move along the nerve. And how does it move along? You know, given the fact we've got this myelin around the outside of some nerve cells, you know, what's going to happen there? All right, so the whole thing is going to be nerve impulse. And the first bit of this is the setting up of what we call a resting potential. Now, that word potential, we've met that a few times recently as well, haven't we? Water, potential, resting potential. So what's potential? It's something that hasn't happened but could, isn't it? You all have immense potential, right? Which is why I've selected you. It's, you know, that's what I look for in students at interview, is potential. You know, have you got the right background? Have you got the right ambitions? Have you got the right attitude? Have you got the right starting qualifications? That sort of thing. That gives you that potential then to move on. So potential is actually something that hasn't quite happened. And with water potential, that is describing the possibility, the future possibility for water to move about, isn't it? So that idea that, you know, osmosis is possible if you've got this difference in water potentials. And with this, the potential we're talking about here is electrical, and it's what we measure in volts. I'm sure you're all familiar with the idea of a voltage on pieces of electrical equipment. And in this country, the voltage we use is 240. Come on. Right, if you travel to Europe, the voltage is... actually, I said 110, yes, I'm not sure that it is. Is it 110 or is it 220? I am not sure. But I've been somewhere where it's 110. And if you take your hairdryer abroad, and it's a UK hairdryer, and you plug it in, it only goes about half the speed. Yeah? Is that Europe? I can't remember now. But because I've travelled to places like Egypt and Turkey and Tunisia and places, I can't remember whether, you know, where the voltage is lower. But if the voltage is lower, what's actually happened then? You know, why is your hairdryer only going half the speed? What that measures, the voltage actually measures sort of a pressure difference in the electrons, in a way. That's a really odd way of looking at it, but it helps you understand why electricity flows. You'll only get electricity flowing if you've got a potential difference, and that's a difference in the amount of electrons in one place and another. And that's what makes electricity flow. You've got to have a difference for it to move from one place to another. And it's like concentration gradients make substances move from the high to the low. The osmotic thing, you get water moving from high to low. So we always get that movement from high to low, don't we? We're going to experience it today in the form of, outside, wind. Yeah, wind. It's going to be very windy, yeah? What's the storm called? Caroline. Yeah, you know that bit. Okay, but it's going to be very windy.
The European electricity is 220.
Oh, it is 220, is it?
Is 110.
America's 110, but I think some other places are as well. So yeah, Europe, I had a feeling. Yesterday, I did say to the other group, I said it was 110, and then I thought, no, it's not. But I know I've experienced 110 somewhere, and I haven't been to America.
Any broker products to 110.
Yeah, I haven't been to America though, but I know I've taken my hairdryer abroad, plugged it in, and it's gone half the speed. So, you know, obviously their appliances are made to work on a lower voltage, but yeah, so it'll be one of the non-European countries. But yeah, we're going to get high winds. Well, I'm not sure if we are, but it's coming in from the north, isn't it? And that's because of a difference in air pressure. So this idea, you know, of potential is a high somewhere and a low somewhere, and you have the potential for things to move. So this idea of the resting potential in a nerve cell is to do with the difference in charge inside and outside the cell, which has the potential for the charge to move and actually switch round, and that's what creates the nerve impulse. All right, so we've got to set up a resting potential. Now, let's just do this in sort of bullet points. There's about 3 of these. They're quite big, but you will need to learn these for the exam. All right, so it's really just a matter of memorising certain aspects of it. Right, so the first thing is that we have this sodium-potassium pump that a lot of you have written about. I have done a quick read through, very quick read through. The sodium-potassium pump is a membrane protein, and it can move both sodium and potassium. Now that's quite unusual, isn't it? If you think about the transport processes you wrote about, the majority of the time you were talking about channels and proteins that could just move one substance. You know, they would be stereospecific for a particular molecule. Like you have a glucose channel, it only takes glucose. You have a sodium channel, it only takes sodium. But the sodium-potassium pump is dual purpose, and it's moving sodium in one direction and potassium in the other. And what it does is to move 3 sodium ions out of the cell in exchange for moving 2 potassium ions in. You're going to have to find a way of remembering that. I will show you a way. All right, now this is active transport, so it's going to need ATP. Yes, it's going to need an energy supply which will change the shape of the protein molecule in order to do the transport. And I'll go back to that McGraw-Hill animation, which we don't necessarily like the accent and the narration, but it's quite short, but it's really, really clear as to how this works. But this isn't going to be enough, so this is only our first step. So first of all then, right, the sodium-potassium pump moves 3 sodium ions out. I'm writing it like that for a very specific reason of the cell, in exchange for 2 potassium ions in. Right? Now, they've both got a single positive charge, but if we're moving 3 out and 2 in, where are we going to have the most positive charge? Outside at this point. So, yeah, that's a start, and this is sort of where we end up. Now, what I should say before I move on is that those are not the only 2 ions involved in this. If you read around this, which of course, you should, but, time doesn't allow, does it really? You will find that this process also includes chloride ions, which are negative, some other organic ions, which might be positive or negative, and it's definitely not just sodium and potassium. But if you understand these, you've got the main part of it. All right. The sodium-potassium pump moves 3 sodium out of the cell in exchange for 2 potassium in by active transport. Now that sets up a difference, a concentration difference for both those ions and an electrical difference. So we're setting up gradients between the inside and the outside that would actually favour them going back the other way, wouldn't they? If you pump sodium out and there's a lot of sodium outside, it'll want to get back in. And if you pump potassium in, you get a lot inside, but it'll want to get back out. That's what we're going to have to look at next. But before we move on to that, I need to give you the way in which you can remember which ones move which way and how many. Okay? Because otherwise, how are you going to remember? How would you remember that it's 3 sodium? And how would you remember that they go out? Equally, how do you know that it's 2 potassium and that they go in? How do you know it's not 3 potassium out and 2 sodium in, or 2 potassium out and 3 sodium? How do you get those numbers all right? I discovered this a few years ago, and it's incredibly easy. But once you know there's sodium and potassium involved, the numbers are 2 and 3, one's going in and one's going out, it does it for you. As long as you know that bit, if I just ask you the two ions, if I ask you which way they move and I ask you how many, you will be able to match them up when I show you how to do it. And I couldn't believe it when I realised how easy it was. Okay, it goes like this with the 3 sodium out. Got it? 3, 3, 3. And you've spotted it. It's 2, 2, 2 for the other one. I couldn't believe that just sort of fell into my lap. And I've not seen that anywhere. It was just looking at it. I'm a great fan of memory tricks. Not necessarily, not tricks in the dishonest way. Tricks in the way of helping you to remember stuff. Creating those nerve pathways is all very well, but then for the potassium, of course, you've got 1, 2, 1, 2, 1, 2. Magic, isn't it? It just sort of, here you are. Here's a ready-made method of remembering this. I think we might have met 1 or 2 other things previously, but not since probably right at the beginning of the course when we were doing things like the characteristics of living things. Do you remember Mrs. Gren? Movement, respiration, sensitivity, those things. And we might have talked about it briefly then, but people have different ways of remembering stuff. And the best way is to find something that works for you. My favourite one is the one for naming planets. Anyone use it?
Well, I use one, yeah.
It's my very easy method, just speeds up naming planets, which was all very well until they took Pluto out of that. But going from the sun outwards, yeah. So because I'm fine as far as Mars, and then after that I get them all muddled up. So I'd have to write it down. And colours of the rainbow. That's it. Richard of York gave battle in vain.
I taught my son to remember how to spell the cause.
Yeah.
My daughter's put big elephants carried down the
Yeah. A lot of people use it for particular spellings that are quite difficult, and wherever you've got a list, it'll lend itself to this sort of thing. I use it on things like postcodes and number plates and things like that as well, just to get the sequencing right. The college postcode is NR32 2 naughty boys, so you get the MB, not BN or something like that, and our postcode is 8TL, so it's 8 of the Leuties. And use it for getting the letters in the right sequence in number plates, that sort of thing as well. And it's all very well when you can remember a number of things, but you can't necessarily get them in the right order. Or if you want to just remember a list of some sort, we will come across other examples, but it is part of what you need to learn, but you have to find your own ways sometimes. So, you might come across a sequence of something that you need to remember. Well, the trick is to pull out individual words, take their initial letters, and then find, something else that starts with those letters that will help you. Very often, the ruder the better. Anyway, that hopefully will help you remember 3 sodium out, 2 potassium in. Now that then sets up concentration differences, gradients, if you like, that are going to favour the diffusion of these ions back in the other direction. So why can't they? Well, they're charged particles. And charged particles can't just get through the phospholipid bilayer, can they? This is, you have just done this. Anybody would think I'd sort of designed this to flow from one thing to the next. And when you were writing about facilitated diffusion, this is the sort of thing that you were looking at, the idea that you can't put sodium ions and potassium ions through the phospholipid bilayer. They are so highly charged, and you've got these non-polar lipid tails that just won't have anything to do with charged particles. They're not going to be able to interact with them, so they can't move through. So you have to have protein channels that have got sort of positive, negative, bits of their own that will allow them through. And there are 2 separate ones. All right, there are sodium channels and potassium channels. But these are not just ordinary channels that let them flow through at any time. They are gated. Gated meaning you can open and close them. So these gated channels can be open or closed depending on, in this case, the voltage across the membrane or the voltage in the cell or whatever. So they are voltage gated channels. Now at this point, all the sodium channels are shut. So there's no way those sodium ions are getting back into the cell. You pump them all out, they cannot get back in. However, some of the potassium channels are open. So some of the potassium can actually leak back out again. Not a huge amount of it, but some of it will leak back out down its own gradient. So you end up with even more positive ions on the outside. So that's actually what's going to give us our resting potential. But let's clarify that. All the sodium ion channels in the membrane are closed at this point, which implies they will open, and they do, but not yet. So sodium ions can't diffuse back down their concentration gradient. I have to put that in because I realise I haven't mentioned it to start with. Alright, so they're left outside. However, some of the potassium channels are open. Right, so some of the potassium channels are open, and some potassium ions leak out. I should have put diffuse rather than leak, I suppose, but they leak out down their gradient, concentration gradient. Now, in the exam, I am likely to give you this as a gap-filling exercise. Alright, so it'll be a written sort of account of what happens with gaps in it that you've got to fill in. So, yeah. However, we've got a lot of revision to do before that. So at this point, we now have a lot of positive ions outside. So there is... Oh, I should have said, sorry, somewhere in there, because I'd said active transport before, what should I have said for this one? Certainly is facilitated diffusion. I'm just going to move that down now. I've taken space at the end. Yes, if you can understand this with the pneumatic drill in the background, you'll be doing very well.
Well, that might help us with it.
Well, it might, but the thing is, yeah, then in the exam you'll need it going again in order to bring it back. That's the
Let's
that's
record it now and then.
it's curious when there is this link between things that occur while you're learning and being able to recall them, and it's the way that memories work. But one of the main things that's being researched is smell. That smell can really be associated with memory quite strongly. Now you might find that if you smell something that you haven't smelt for a long time, you'll immediately think of an association. And it can be things like, the smell of a school, or the smell of a hospital, or the smell of, an elderly relative's perfume, or something like that, will really bring memories back. And you know, they can be quite vivid things and things from quite a long time ago. But there's been quite a lot of research into it, and there's a reason for this, and it's because where the olfactory nerve goes through your brain, that's your smell nerve, is very close to the part of the brain that deals with memory. So stimulating the sense of smell while you're forming memories can actually be quite helpful. Now, I used to teach aromatherapists, and they would swear by this, and particularly the smell of rosemary is supposed to actually stimulate memory production. So, you know, when you're revising, get some, have some rosemary about, or rosemary essential oils in a burner or something like that. The only problem is, in order to make it work really well, you actually need to recreate the smell when you're trying to recall it. So you could use any smell. I mean, you could, but then, you know, you go into the exam, if everybody's got a different smell, it's going to get quite revolting. But if you I mean, or you can use something, you know, just that you would agree on, not something like lemon or mint or something like that. That's, you know, not not going to be a problem, but that might just help you to bring back. But you've got to use it while you're revising, and then, you know, use it as a recall thing. But it is, you know, it is a genuine thing. All right, this is not just made up. However, the sound of a pneumatic drill in the background is probably not going to help. Right, where was I? Now, need to describe what we've got. So we've got a lot of positive ions outside. Now, this is ignoring all the others, of course, but this is positive ions outside compared with the amount inside. Now, the way that we measure the membrane potential is we say, what's the inside like compared to the outside? So the inside is described as having a negative charge and the outside as a positive charge, and the difference across the membrane is described as being minus 70 millivolts. So the inside has sort of 70 millivolts less charge than the outside. It's not really that the inside is perhaps negative, it's just that if you compare the inside and outside, so it's this difference in charge. Now, at this point, we can describe the membrane using a word that we've described the water molecule as. How would you describe its positive end and its negative end? Makes it polar. Yeah, so the membrane at this point, being positive outside the membrane, negative inside the membrane, we can describe the membrane as being polarised. All right, these words do come up over and over again. They're not just found in one place. That's our resting potential then. It's minus 70 millivolts, the membrane's polarised, and it's ready for action. So let's finish this bit off. So there are many more positive ions outside the cell than inside. The outside is described as positive, and the inside as negative. I'm not going to say it, but I'm not going to write it, but you know, this is a relative thing. It's not an absolute sort of charge thing. And the membrane is polarised. Okay, so that means we've got the two extremes, the positive and negative. So we've now got our resting potential. You will find slightly different values for this depending on where you look, but the resting potential is, we'll say, approximately. Not all nerve cells are equal, but it's about minus 70 millivolts. The minus is critical. If you don't put the minus, you are wrong, right? So very, very important that that's there. So this is a cell that's not conducting an impulse. This is a cell that's just waiting for some sort of stimulus. And what I'm going to do now is to draw a membrane with some numbers to show you how you can actually get a minus 70 at the end by these processes. But these are completely arbitrary numbers that I've made up just to illustrate this. So, this is a nerve cell, a fibre, an axon, a dendron, doesn't really matter. And well, that's straighter than I drew it yesterday. Alright, so this is the inside of the neurone. And this is the outside. Oh no, hang on a minute, I need space to put other stuff there. Just assume I've written that. Right, now I'm going to use two colours for the two ions. Outside the cell I'm starting with 100 sodium ions, and inside I'm also starting with 100 sodium ions. Outside I'm starting with 100 potassium ions. Inside I'm also starting with 100 potassium ions. So the charge inside and outside at this point is equal. All right, so the total charge outside is 200, yeah, same inside. So there's no difference, there's no potential difference there, there's no voltage at this point because you've got the same charge inside and outside, so the charge isn't going to move, the ions are not going to move, not of their own free will, not that they have any free will anyway, but they are not going to move on their own. So the first thing that we need is our sodium-potassium pump. And we're going to exchange 3 sodium out for 2 potassium in. But I'm going to multiply that up a bit. And I'm going to move 60 sodiums out. I think. So what we're going to do, 60 sodium going out of the cell. In which case, how many potassiums are going to go in? 40, yes. All right, for our 3:2 ratio, if 60 Na pluses go out, then we must have 40 potassium ions going in. That is carried out by this one protein, the sodium-potassium pump. So this is our active transport. So that's what's happening here. And so we now need to check on the number of ions that we've got inside and outside again. So let's look at our sodium ions. How many have we got outside now? 160. How many have we got inside? 40. So we've got a heck of a gradient there that would encourage the sodium to diffuse back in again, but you know, we're going to find that sodium channels are shut, so they can't get back in. So we're left with our 160 outside at this point. Same sort of approach with our potassium. So how many have we got outside now? 60. And how many have we got inside? 140. So we have already changed the charge inside and outside. It just isn't enough though, see. So what's our total charge outside now? 220, and inside 180. So there's a big difference already, but it goes further than this. The next step then is to look at these ion channels, the gated ion channels. The sodium channel at this point is closed, so it will not let any sodium in. So here's our sodium channel, but it's closed. So no sodium getting in. Now, you've probably seen the diagrams when you're doing your assignment, the little channels with like a little flap on them that could be closed, but we've got some good diagrams in the handouts in a bit anyway. The potassium channels, on the other hand, are not all open. There's some open. So I'm going to show that as a dashed line because it's not really very great, but we get, to make this work, 15 potassium ions diffusing back out through their channels. Okay. So let's add up our numbers. Sodium-wise, what do we got outside? Now, just sodium. Still got the 160, haven't we? Because that had to stay there. What we got inside? Still got 40. But we've had a change in the potassium. So what we got outside for potassium? 75. Not really left enough space here. And what have we got inside? 125. So let's do some adding up here. What's our total charge outside? 235. What's our total charge inside? 165. So how would you describe the inside compared to the outside? Yeah. By how much? 70. And that's our minus 70. So the difference from here to here, that's my minus 70 millivolts, if you like. All right, so that's just to show you how these two processes combine, the active transport and the facilitated diffusion combine to give you a difference that we describe in volts. You know, I mean, positive charges are not volts, but it's the difference in the charge. So I just picked numbers that would sort of help a little bit. I'm going to just show you that. Oh, I know what I need to put on there. I haven't. I put active transport under that. We can have facilitated diffusion under this. All right. Now, there are animations out there, and if you need to see this in a, you know, an animated sort of way, then you need to do a bit of hunting around. I couldn't find one quickly yesterday, but I will come back to this if you're still writing that down. I'm just going to find the active transport animation. I'm going to use the one that I've used before rather than these ones. Going to use that one because I know that it does what I want it to do. There's loads of others, so please use them. This one, all right, just a reminder that this is what's happening at that first step, and then you have to have the facilitated diffusion.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay, so that's our, that's the active transport bit. That's the sodium-potassium pump. I noticed up here on their list there is one for facilitated diffusion. I'm just going to give that a quick look. It may or may not be helpful. I'm not sure if I've used it. Oh, we have used it. It does sort of give you an idea of the potential for it being gated, because these little sort of flap things at the top and bottom could close. Let's just have a quick look.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid.
Or an ion.
Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
Okay, well, that's fine. It doesn't look at the gated nature of the sodium and potassium channels that we want, but that'll do for now anyway. So at this point then, we've created our polarized membrane. We've got it effectively positive on the outside, negative on the inside. But that, like I say, that's relative. Okay. So what happens then, when you want a nerve cell to actually transmit an impulse, what's this got to do with it? Well, because we've now got this difference inside and out, you can actually change that, and it switches round. And just an overview of what happens next is, well, you get what's called an action potential. And then action potential moving along the neuron is an impulse. Now, the action potential is described as being when the membrane is depolarized, but actually it sort of gets polarized in the other direction. You go from polarized at this point, the resting potential, depolarized at the peak of an action potential, and then it has to be repolarized to get back to its resting potential. Now that whole cycle takes milliseconds. We're talking about 3 milliseconds probably for the complete process. 3/1000ths of a second, all right, to do this. So you think about all the processes that are happening there, it's incredible. Right, so our next job then is to have a look at what an action potential is, to consider how it actually moves along a neuron, and to look at a few of the factors that might affect how fast it moves. There are 3 factors that affect speed of nerve impulses. One of the important things to understand, I'll say it before we start, is that you either get an impulse or you don't. They do not vary in size. They vary in frequency along a neuron, but they don't vary in size. So you can't have a large action potential and a small action potential. You either get one or you don't. And you might have very frequent ones, or you might have quite less frequent ones. And it's that determines the intensity of what you feel or how strong the response is. Right, so this is the other part of the story then, the action potential. The first thing that we need is some sort of a stimulus, and a stimulus could be all sorts of things. It might be an impulse from another neuron, it might be light shining on a receptor in the eye, it might be touch, it might be a chemical, but some sort of stimulus. Now, the way that stimulus affects the cell is actually quite difficult to explain. The easiest one to explain is actually in the eye, because light falling on the rods and cones in the eye actually creates a chemical change that changes the ions inside the cell, and that triggers what we call a generator potential. With a lot of the others, it is really quite hard to explain. But we don't need to be able to, as long as we understand that a stimulus is required to start with. Now, that stimulus has to reach a certain level. We call it a threshold level. Once it's reached the threshold level, it will then trigger the action potential. Now, that level is sometimes also called the liminal level. So if a stimulus doesn't reach a threshold level, how might we describe it? You'll know the word. If it reaches, we've got two words. Threshold level is also known as the liminal. What does that? What? No, liminal. Have you heard of subliminal? Yeah, yeah, oh yeah, yeah. Subliminal is something that you're not really aware of, isn't it? Subliminal messaging and things like that, you know, and some subliminal information is sort of below the level of stimulus, so you're not really aware of it. All right, so the liminal level is the threshold level. I tend not to use that term though. So once the stimulus has reached the threshold, that will then trigger the action potential. What happens? Well, remember those sodium channels were all closed. All of a sudden they start flying open. That's what the stimulus to threshold level does. It starts the sodium channels opening. So these gated channels start opening up. The sodium ions rush into the neuron, because that difference, go back to my numbers, that gradient was quite significant. You know, we've got 160 outside, 40 inside. They desperately want to get back in. So if you now open these channels where I've drawn the cross there, they are going to rush in very, very readily. And as they start rushing in, because that then starts to change the voltage, that makes more channels open, more rush in, that makes more channels open, that makes, you know, and so on. And that is a feedback process, but it's a particular type of feedback. Positive. Yeah? It's positive feedback. We talked about negative feedback being something that stops a process. Positive feedback is one that encourages it to go on further. And this is one of, I said there'll be two examples of positive feedback. One is in this, and of course the other one is in labour, where, you know, pains and contractions build until you know, the actual birth process puts an end to it. With this, the positive feedback goes to the point where the membrane's fully depolarized and the channels will then shut again, but we'll come back to that in a minute. So that's our first important bit then, is that stimulus to threshold level causes the sodium channels to start opening. And sodium ions rush in. Now we need to put the positive feedback in here. As more sodium ions move in, more channels open. And this is positive feedback. And then I will say until the membrane is depolarized. And what we now do is compare the inside with the outside, and we find that the inside's positive compared to the outside. And the value you get for this does vary depending on your source of information. But I would usually say somewhere between plus 40 and plus 50 millivolts. And this is our action potential. So it's somewhere in the order of plus 40 to 50 millivolts. So we've got some important words in there. We've got positive feedback, we've got depolarized, we've got action potential, we've got a value. So there's quite a lot in that one bullet point. All right, now at this point you can't just stop. So what happens now? Well, almost immediately, and we're talking sort of within about a millisecond, those sodium channels shut again. Because they've reached this point, and that then triggers them to shut. They're voltage gated, so when they reach that point, they close. But as they do so, the potassium channels now all fly open. Remember, there were only a few open before, so they all open, and the potassium, of course, can now rush which way? Out. That's it. So that puts more positive ions back on the outside, and it actually repolarizes the membrane. All right, so that's our next bit. We get this repolarizing. There is a bit of an overshoot, and we've got to explain what happens at the end, but this is the next step. So almost immediately, the sodium channels close, and all the potassium channels open. So potassium ions flood out, repolarizing the membrane. The trouble is, this is not repolarized to the point that's actually going to be any use at the moment, because they will only flood until you've got equal concentrations, won't they, on each side? Although what tends to happen with this is that the potassium is so eager to get out, it overshoots somewhat. And it actually overshoots past minus 70. Now, the thing about that is it has to be sort of set back to minus 70, so the active transport start up again. But there is a sort of useful side to that, because it does mean that you can't then send another impulse down that nerve too quickly. It's got to wait till it's reset. So we get an overshoot that we describe as hyperpolarization at this point. So I can't think of a better word, so I always put it in quotes, because overshoot is not really perhaps the correct way of saying it. But the overshoot causes hyperpolarization, and then we're back to square one here, where the sodium potassium pump starts to restore the resting potential. Now, while it's doing that, the membrane can't respond. And this little period of time between the end of the action potential and restoring the resting potential, it's called the refractory period. It's like a resting period. And the first part of that refractory period is absolute. You can't get the membrane to respond no matter what you do. You know, your ions are all in the wrong place, so it doesn't matter how hard you poke it, it's just not going to react. However, once the sodium-potassium pump has started to restore the resting potential, a really strong stimulus will actually generate an impulse. Now, I have my analogy for this is working less and less well as the years go by due to plumbing technology. Because my analogy involves toilet flushers. And the trouble is, this only really works for toilet flushers that involve a handle or a pull chain. Doesn't work for button flushers. All right, now you've got to think about, all right, toilets being flushed. We will not go into any reasons why, but let's assume that we want to flush it a second time quite quickly. These things happen. If you want to give it a second flush, you know, you might have just blown your nose, chucked another bit of tissue down the toilet and just want to flush it before you leave. If you want to get a second flush out of it, you can't do it straight away, can you? You know, think about the handles and the pull chains. You must have come across them somewhere. You know, if you try, once the cistern's empty, you try and flush it again, nothing happens, does it? You just hear that empty cistern sound. However, if you let it fill up just a little way and you go whoosh, 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 you can get another small flush, can't you? And that's what the nerve impulse is like here. That you get that absolute refractory period, think empty cistern, not going to happen. You wait sort of 20, 30 seconds and, you know, cistern's partly filled or the membrane is partly reset. And if you really, you know, smack it hard, it will then respond, right? So, yeah, I'm never really convinced about the use of that analogy, but yeah, there it is. So this is... So this is the refractory period. And we've got two bits to it. Right, you have the absolute bit. Membrane totally non-responsive. And then you've got the relative, where membrane responds to strong stimulus. All right, so what you can get out of this is, as I said, all or nothing. So I now need to, I don't know whether this is another bullet point, let's go for it anyway. So movement of action potential, and we've got a handout to look at that goes with this. I've just left it all completely till the end. So movement of action potential along neurone equals the impulse. I should have just called it the AP, shouldn't I? Look, AP for action potential is, it's an all or nothing event. It only goes in one direction as well, and that's maintained by this need to repolarize the membrane. So, you know, if you want nerve impulses in two directions, you've got to have two neurons. It can only go one way. Before we look at the factors that affect the speed of an impulse, In your handouts you have a diagram which has got this, it says action potential at the top corner, top left corner. And then you've got this graphy thing across the middle. Alright, so you've got a thing that goes like that, and then like that, and then peaks and it comes down, and it goes down, and then comes back again. And that's effectively the electrical trace that you would get with a nerve cell being stimulated. And what you'll see with this is that you've got the label boxes around the outside with label lines going to each part of this trace. Now, it's actually quite useful, but the problem with handouts, and I'm very wary of giving handouts, but I have to, really. The problem is they can just be pieces of paper that you have. You know, I've got handouts. Well, whoopee-doo. You know, I've got an exercise bike. Doesn't mean I use it. All right. So, you know, using handouts is the important thing. They provide you with a lot of the sort of additional explanation I don't necessarily have time for, or, you know, reiterating things that I've said, or a slightly different version, or what have you. So it's really quite useful to learn how to use them. This particular style I quite like. This is out of an A-level revision book. And you have these lines and their boxes, and it works quite well on this one if you highlight the label line, the box, and the bit of the graph it applies to. So, for example, I'm going to just go with a bit over the side. Over here, where the graph line goes up, your box is over here, and it says that represents the depolarization bit, where the voltage-gated sodium channels open, the sodium ions move into the axon, down the sodium concentration gradient, down the electrical gradient. You've got an image there of the sodium channel opening, but the potassium channel's closed. But it's linked to that part of the graph where it goes up. So, right. Tend to go over the top. Well, let's use that anyway. That what I would be tempted to do would be this. That box refers to that bit of the line, yeah? If that doesn't work for you, don't worry. You don't have to do it. This is not compulsory, but this is just, you know, trying to find ways of helping you study and learn. Now, once you reach the top of that, of course, you come over the hill and down the other side, and the box for that is sort of over on the right hand side, not the completely top one. The one that's going across the top is a little bit harder to highlight because it's fitted in right up to, it's a bracket. I don't know whether I can do that. I'll give it a go in a minute. I might have to use a smaller highlighter. But you've got this bit, which is the repolarization described over here, where the sodium channels have closed. You can see that, but the potassium channels open, so they're getting out again. So if I highlight that with, let's go over here, like that, and that's the bit down here. You then have a bit down at the bottom for hyperpolarization and the refractory period. All right, so that, I'm just going to go for just sort of there. All right, that's the bit down here. Oh, I'm not putting all the boxes in now. The threshold level is the dotted line going across here that just shows, you know, you've got to reach this point before it'll actually shoot off. So I have got one left, haven't I? A pink one. Pinky. So that one can go. That could actually be across the. Oh dear, that's messy. That was an interesting effect, wasn't it? Yeah, colouring. Yeah. But the bit that we're missing a label for is up at the top. It tells you about the action potential. Now, what you've got, and I've really run out of pen options here. I want to, hang on. You may only just be able to see it, but you've got a bracket that's taking this bit. Not quite sure. Yeah, it goes to there. And then it goes up to the top, doesn't it? Whoops. You've got a box running along the top. Yeah? If you've got a small highlighter, so I was like, alright, I'm going to use, if I use, trying to find one that doesn't, no. If I go with the yellow, then that bracket is up here. You might find that's made a hideous mess and you wish you hadn't done it. Sadly, no. I've got an unsullied one if anybody wants to copy it. But it makes the graph more meaningful to link the label boxes with what's going on. Honestly, it does. And it shows you those channels. You can see in that hyperpolarization one, both the channels are closed at that point because the only thing left working there, of course, is the sodium-potassium pump. And they're not illustrating that. So, yeah, if that got a bit messy, I'm sorry about that. But it's just, you know, it's partially an exercise in helping you to use resources. Now, on the other side of this sheet, you've got a little bit about how action potentials move along a neurone.
Oh, I see. Yeah.
The top diagram, I think, is just showing a piece of membrane. The bottom diagram is showing myelinated. So, if you've got, let's say you've got a non-myelinated neuron, you've got, you can do. This would be non-myelinated. You've got to sort of, I'd have to have the same all the way along here. Right, if in a non-myelinated neuron, your action potential would have to work its way along, whoops, that wasn't right, it would have to work its way along every little bit of surface. So it would be sort of all the way along. And that's quite hard work. Takes a lot of energy. Requires a lot of depolarizing. What you've got to remember is, it's the polarizing that takes the hard work, doesn't it? Because that's the action potential, the active transport. If, on the other hand, you've got a myelinated neuron, you cannot move sodium and potassium through the bits of membrane where the myelin is. So, you might have the action potential will just exist here. And so you get it jumping. Right? It's jumped from there to there. Then it jumps from there to there. It's not doing every little bit of the membrane. So that's non-myelinated, that's myelinated. So what do you think the effect of myelin is on the speed of a nerve impulse? Makes it a lot quicker, yeah. It would be as if I was trying to walk the length, or let's say the width of this room, just going heel to toe, you know. Heel to toe, heel to toe. I've got to touch every bit of floor, I've got to depolarise every bit of floor, you know. It takes a while to do every bit. On the other hand, if I just have to make contact with little bits, I can stride across and it's a lot quicker, it's a lot more energy efficient. So the presence of myelin will speed up nerve impulses. There's a couple of other things, one of which you should be able to make an intelligent guess at. Generally speaking, in processes that I just realised I walked away from the transmitter, so that anyone who's listening to this will not have just got me walking across the room. So the presence of myelin will speed up a nerve impulse. Something else that will speed up processes generally. Just a physical factor. So, yes, temperature. Yeah. So in warm conditions, the impulse will move faster. So what impact will that have on animals broadly? Are all animals equal body temperature? No, we have, yeah, cold-blooded and warm-blooded, which is a very, so that's not terribly accurate, but mammals and birds are warm-blooded, so their nerve impulses will be faster. Reptiles, amphibians, fish and invertebrates are what we call cold-blooded. They rely on the temperature of the surroundings to warm them up. So until they're warmed up, their nerve responses will be slow. Once they're warmed up, they'll be fine, but temperature. Now the other thing, your instincts will usually sort of, confuse you on this one. The other thing that affects the speed of a nerve impulse is the diameter of the nerve fibre, diameter of the cell. So if we've got a bigger nerve fibre, you know, that far across, let's say, instead of that far across, the bigger nerve fibre, do you think it'll conduct more quickly or more slowly?
Slowly.
It's interesting, everybody always says that, but it conducts more quickly. The reason being that you've got less membrane for the amount of cell. The cell produces energy. So in a big cell, you've got loads and loads of energy being produced for that membrane. Whereas if you've got a lot of little cells, I'll draw this, or I'll try and show you anyway. Hang on, let's do it. Let's keep track of this. Oh, incidentally, when you go for break, don't forget you've got cake. Those who are not here haven't got cake. Yes, and they say thank you, Sam. Right, the factors affecting the speed of the nerve impulse. It's faster if, first of all, we've got if it's myelinated. Secondly, if it's warmer. And then thirdly, if it's a larger diameter. And this is the one that I just need to explain before we stop. If you've got, here's a great big nerve fibre, and here's the equivalent. Oh, I wish I'd done those a bit bigger. Let's assume, you know, I was going to fill that in with small ones. Now, this amount of cell here with its mitochondria has just got to polarize and depolarize this much membrane, yeah? The same amount of cytoplasm in the other one, in this setup, got the same amount of cytoplasm, yes? The same number of mitochondria. It's got a lot more membrane, hasn't it, to work on. Does that now sort of make sense? It's one of those situations where a small surface area to volume ratio is actually better. So it's smaller surface area to volume ratio for membrane to contents of the cell. That means, you know, which includes cytoplasm and mitochondria. Okay. Right, well that is it's a very convenient point to stop and take a break. After break, and cake, yeah, don't forget your cake, we will come back and we'll do a little investigation to see how fast your nerve impulses are working this morning. So be interesting, you know, see who's had caffeine, who's had nicotine, who's had a little nap. Alright, so we will, yeah, we'll do that. We will do that in a bit. I think I can stop this at this point.